In this video, we're going to talk about liver metabolism. And I know that's not really a topic, liver metabolism, but like all different metabolisms that we talked about like glycolysis, Tca cycle, that we talked about in the muscles and in the blood. We're going to discuss it all occurring in the liver and how different enzymes and substrates react to our body when we're hungry or if we're fed. So the fed state or the non-fed state. Now, I know this looks crazy, which it kind of is, but I'm going to break it down as simple as possible. I looked for YouTube videos online, and there really wasn't one that like talked about this whole entire diagram, so I kind of like made my own the best I could. And I organized it not in like a flow order, I organized it like in a systematic order. So like the Tca cycle is going to interact with these systems. Glycolysis is going to interact with these systems, right? It's not just like a descending order. It's just like the order that makes most sense in my head. So, if we're going to start off in glycolysis. Now, we've spoken about glycolysis multiple times. You know what it is. But it's important to do a quick rerun through it because there's all these different pathways are going to be involved and are incorporated in it. Now, how does glucose get into our body? We know glucose goes through the blood through GLUT4 transporters. And now if we're going into the muscle, or into the liver, the muscle is going to be a GLUT4 transporter. If we're going through the liver, we're going to be a GLUT2 transporter. Now, why am I saying blood to liver? If you see this board, it kind of looks like it's boxed in, like this whole line going across, down the side, etc. The box is representing basically like muscles and livers, and outside of the borders is going to be the blood. Just a quick like tidbit. So now the glucose is going to enter in, right? And then we're going to use an enzyme called glucokinase. For glycolysis, we usually use hexokinase if we're talking about muscles, right? You have hexokinase or glucokinase. We have a higher affinity for glucokinase in the liver, so glucokinase is going to be used. Now, in this step, we have to give energy, so ATP is going to get converted to ADP. And then we're going to turn glucose into glucose 1-phosphate. That's step one of this whole process. Not too bad. Now, I have red lines going in all different directions, right? In the beginning, this red line is going to represent enzymes or substrates that are coming from different reactions that are being incorporated into this cycle. So, glucose to glucose 1-phosphate. We're also going to see this glucose 1-phosphate in the lactose metabolism. So, metabolizing galactose is milk, dairy products, cheese, etc. So just know you're going to see these enzymes a lot. That's why there's overlap. But now, if we go from glucose 1-phosphate to glucose 6-phosphate, we're going to use an enzyme called phosphoglucomutase. Glucose 6-phosphate, as you see, is used in a lot of reactions. We're going to be involved in gluconeogenesis. It's going to be talked about again in fatty acid synthesis. A lot of different pathways, which we'll get to in a little bit. But just for now, just know glucose 6-phosphate is coming from glucose 1-phosphate. Now, after that, glucose 6-phosphate becomes fructose 6-phosphate via phosphoglucose isomerase. This is a reversible reaction, so it could go down or it could go up. All these are, right? This is a reversible reaction. This guy is not, though. But this is reversible. How is it reversible? We'll talk about it in a little bit, but just for now, just know that. Now, fructose 6-phosphate becomes fructose 1,6-bisphosphate via PFK1, which is phosphofructokinase 1. We know in glycolysis that phosphofructase kinase 1 is in tangent with phosphofructokinase 2, right? They're allosterically inhibited by each other. PFK2 could turn on PFK1 or turn it off, etc. But just know PFK1 controls the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Now, these two can split, or fructose 1,6-bisphosphate can split in half by an enzyme called aldolase. Aldolase divides it into glyceraldehyde 3-phosphate or dihydroxy acetone phosphate, also known as DHAP. DHAP is going to be used a lot, especially in gluconeogenesis, but just know for now, we get glyceraldehyde 3-phosphate and DHAP, dihydroxy acetone phosphate. Now, how did we get to this? How do these guys switch off? Because you know these two substrates are interchangeable. There's an enzyme called triose phosphate isomerase, which flips them back and forth. Now, glyceraldehyde 3-phosphate can continue down through glycolysis. The next enzyme or the next substrate it goes to is 1,3-bisphosphoglycerate. Okay, how did this happen? We used an enzyme called glyceraldehyde 3-phosphate dehydrogenase. Okay, if you see dehydrogenase anywhere, you have to know that it's usually taking an NAD or an NADH and it's converting it. So we're going to use something, right? We're going to use a nicotinamide adenosine substrate to help with this intermediate reaction. Now, it took NAD and it became NADH. Let's remember that one. Now, 1,3-bisphosphoglycerate is then converted into 3-phosphoglycerate, also known as 3-PG, via phosphoglycerate kinase. That's going to be the enzyme that uses that one. Now, 3-PG could go to 2-PG, which is 2-phosphoglycerate. This enzyme is phosphoglycerate mutase. After 2-PG, we go down to phosphoenolpyruvate. This is another important step. Why is this going to be another important step? This step is going to be used, or it's going to be our first step used in gluconeogenesis, which we'll get to in a little bit. Now, 2-PG to phosphoenolpyruvate uses the enzyme enolase. From phosphoenolpyruvate, we go to our last substrate in glycolysis, pyruvate. Why is pyruvate so important? Pyruvate is going to be involved in a lot of different systems, and it's going to be converted into acetyl CoA or lactic acid. Now, how do we get from phosphoenolpyruvate to pyruvate? We use pyruvate kinase. That's the enzyme that's going to do that. Okay, so what did I say? Pyruvate can be converted into lactic acid, which is in the muscles, right? Our muscles are going to use this. Or pyruvate could go to acetyl CoA via pyruvate dehydrogenase. I said dehydrogenase because you use one of these things, NADH to NAD+. What's important about this reaction is that up here, we went from NAD to NADH. Down here, for the lactic acid, right, we're going from NAD+ to NADH to NAD+. This NAD+ is then going to be recycled where? Back at this glyceraldehyde 3-phosphate to the 1,3-bisphosphoglycerate step. Okay, so that's for both lactic acid and it's for acetyl CoA. Now, acetyl CoA goes to the Tca cycle, which is also known as the Krebs cycle. That's going to be this whole entire diagram right here, which we're going to get to. But for now, just know, glycolysis, we took glucose, so one sugar molecule, went through this whole entire reaction and generated a net of 2 ATPs, 2 adenosine triphosphates, which is going to be used for energy. This is known as substrate level phosphorylation. It's quick, it's fast, and it's effective. However, we don't have that many stores of it. Real quick, our energy systems. The fastest energy system we have is our creatine phosphate energy system. It lasts from 0 to 15 seconds, right? It's immediate energy. After that 15 seconds, we switch into glucose. How am I going to explain this? Think of running a 400-meter race, right? A 400-meter sprint. The gun goes off. The first 15 seconds, you're using the creatine phosphate pathway. After that 15 seconds, you can't sprint full speed anymore, right? You're like, all right, I'm still moving pretty quick, but I'm slowing down. Like, I'm running out of steam. From 15 seconds to about 50 seconds, we're going to use glycolysis. This is going to be our substrate level phosphorylation. We're like, boom, 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 make an ATP, make an ATP. After you start burning out of glucose, we need to make energy from somewhere else. Well, we're going to make this energy via the Tca cycle. There's going to be other pathways which we're going to talk about. But the next path we're going to get into is the Tca cycle, which is going to make energy slower, right? But it's oxidative. So it's more energy, but at a slower rate. That's why after you run your 50 seconds, right? You start gassing out. The last 30 meters of that sprint, it's like mind over matter, they say. You're trying to fight your fatigue because you're trying to fight against these two systems. Anyways, that's everything for glycolysis, which was a lot, but it's going to make, I did this video long because I want to make every other system, like, be intertwined a lot easier. Glycolysis, now we're going to work with the lactose metabolism. I mentioned before that glucose 1-phosphate is involved in the glucose pathway, right? It's one of the intermediates. Well now, say we're eating, right? And we don't just ingest glucose, we drink milk, lactose. Lactose is a form of sugar, right? It's a disaccharide. Well, it's a disaccharide. Now, how can this disaccharide be used for energy? All right, so lactose is going to be converted via an enzyme known as lactase into glucose and galactose. Glucose and galactose are just monosaccharides. Now glucose, right, it can be reused, well not reused, it can be used through glycolysis. Right? This whole pathway we spoke about right here. But what about the galactose? How's that going to be used? Is it just going to be excreted out of our body? No, we're going to use it as energy. How? Galactose is then going to be converted into two different substrates or intermediates. It's going to go to galacitol via aldose reductase, or it's going to go to galactose 1-phosphate, which is galactokinase. Now, galacto 1-phosphate and glucose 1-phosphate are kind of the same. How do you make this, or how does our body make this galactose 1-phosphate into glucose 1-phosphate? It uses something known as UDP glucose, and UDP glucose, all it is a substrate kind of that is incorporated into this galactokinase 1-phosphate uridyl transferase enzyme, and it's just transferring something. So it's transferring UDP glucose into UDP galactose, and then the result is a glucose 1-phosphate. Now, this glucose 1-phosphate, as you see I have written right here, goes into glycolysis or gluconeogenesis. As you see in glycolysis right here, the pathway or the substrate, the intermediate, is right here, glucose 1-phosphate. So now, if you were to mesh all of these things together, like these whole pathways, because that's how our body has it, glucose 1-phosphate would fit right into here into the puzzle piece, and everything would be meshed around next to it. And then this glucose would be reabsorbed or reused up here. So that's everything for galactose metabolism. Now, the next thing I want to talk about before I go into fructose metabolism, so if we're drinking like a fruit juice or something. Now, the pentose phosphate pathway, what is this used for? Well, it's going to help regulate a lot of things. It's going to help us make NADPH, which is going to be used for biosynthesis of our fat. It's going to be used in creating ribose 5-phosphates. We're going to make nucleotides, nucleic acids, coenzymes. It's going to help regulate cancer. This pentose phosphate pathway is very important. However, there's a couple things that we should know about it that's going to help incorporate into glycolysis. So it's also going to go into our system. Now, glycolysis or gluconeogenesis, I should say, because this pathway is going to be used mainly for both of them. What two intermediates are we going to form in the pentose phosphate pathway? We're going to form fructose 6-phosphate, and we're going to form glyceraldehyde 3-phosphate. Okay, so if we're forming glyceraldehyde 3-phosphate, where else do you think we would see this pentose phosphate pathway being incorporated? Right into here, into the fructose metabolism. So just see, I can't draw all these arrows, but just see how much all this stuff goes hand in hand with each other. It's nuts. Now, let's start. So glucose is going to come into our liver. This is the liver. Remember, this is all the liver. Glucose is going to come in our body via a GLUT2 transporter. Glucose is going to be converted into glucose 1-phosphate. Glucose 1-phosphate becomes what? Glucose 6-phosphate. What substrate or what enzyme does this? The same one we saw up here in glycolysis. Glucokinase. Now, glucose 6-phosphate is going to be converted into 6-phosphoglucanolactone. What enzyme? Glucose 6-phosphate dehydrogenase. Dehydrogenase, what does this mean? We see NADP plus to NADPH. These is going to be very, very, very important, but in fatty acid synthesis, but we'll get there. Now, 6-phosphoglucanolactone is going to be converted into 6-phosphoglucanate via lactinase, and it's going to take a water molecule and make it an oxygen molecule, so it's going to use a hydrogen. Now, this 6-phosphoglucanate is going to be converted to ribulose 5-phosphate. This ribulose 5-phosphate was made via 6-phosphoglucanate dehydrogenase. Here we go. We see dehydrogenase. You have NADP plus to NADPH. CO2 is also released here. Just note that. Now, one more. So just to note, from here, this glucose 1-phosphate, all the way down to the last step we're going to talk about is going to be known as the oxidative pathway of the pentose phosphate pathway. And it's going to make NADHs and ribosome, like ribose phosphates. Now, this ribulose 5-phosphate could go two ways. One way, it could be converted to xylulose 5-phosphate via epimerase, or ribose 5-phosphate and isomerase. Okay, so we have two different pathways, right? We have a xylulose 5-phosphate and a ribose 5-phosphate. Now, there's these two enzymes, or these two intermediates, I apologize, are going to join together, and they're going to be broken into two different regions. This is going to be the start of the non-oxidative pathway. So we're going to have TPP, which we saw in purine pyrimidine metabolism, and that used vitamin B1 with the enzyme transketolase, and we're going to make two different structures. We're going to make ceduloheptose 7-phosphate and glyceraldehyde 3-phosphate. Here we go. Glyceraldehyde 3-phosphate is made. It's getting reincorporated into glycolysis or into fructose metabolism. So that's our first thing we got. Now, say we're not using this, right? This glyceraldehyde 3-phosphate and this ceduloheptose 7-phosphate are going to join together via transaldolase, and they're going to make two different byproducts. They're going to make erythrose 4-phosphate or fructose 6-phosphate. Well, here we go again. Where is this fructose 6-phosphate going to be involved? He's going to be in glycolysis again. We have fructose 6-phosphate being made as a byproduct of the pentose phosphate pathway, the non-oxidative region. We're going into glycolysis again. Now, this erythrose 4-phosphate, right? He's going to join with xylulose 5-phosphate, which came from this reaction up here. So this xylulose 5-phosphate is going to join with erythrose 4-phosphate via transketolase, same exact enzyme used here with vitamin B1 and TPP, and it's going to form glyceraldehyde 3-phosphate and fructose 6-phosphate, which are going to go to the same places we just spoke about before. Now, those places are where? Glycolysis and fructose metabolism. We know this. Now, this is a non-oxidative pathway. So what does this mean? This means we can make ribose 5-phosphate, so we can make this product right here, right, without making NADH. How? We're going to take these glycolytic intermediates, because this is a reversible pathway. We're going to take these intermediates, send them down through the chain, work backwards. So if they're working backwards, they're going to make ribose 5-phosphate. So we can make this working reversed. Or, say we don't have these glycolytic, we don't have glucose, right? We can make these glycolytic enzymes. How? Well, we could go from ribose 5-phosphate, make these byproducts, or make these intermediates, and then send them up the chain to glycolysis. So everything's working hand in hand with each other. So that's basically the whole pentose 5-phosphate pathway. And just know the main thing we want to talk about is the creation of NADPH. This is going to be really important for our fatty acid synthesis. The Tca cycle, right? Where is he getting his supply from? I just said you were running a sprint, 15 seconds creatine, up to 50 seconds glycolysis. Now you're starting to fatigue, right? You want to run a mile, 2 miles. You're going to use a Tca cycle. Pyruvate is getting converted into acetyl-CoA, so we're going to see it right here. Pyruvate to acetyl-CoA via pyruvate dehydrogenase. We said the same thing over here, pyruvate dehydrogenase. If it's dehydrogenase, you go from NADP to NADH, or NAD positive to NADPH or NADH, which is then going to be used later on in the electron transport chain. We'll get to there. But now, pyruvate dehydrogenase, right? Makes pyruvate to acetyl-CoA. Now, this is where things get very important. Oxaloacetate, which is the last step of the Tca cycle. So the last step is going to be involved in forming the first step. So the last step, oxaloacetate, is going to join with acetyl-CoA, and it's going to form citrate via citrate synthase. Okay, so we're forming citrate via citrate synthase. Okay, oxaloacetate, acetyl-CoA, citrate synthase. Now, citrate is then going to form isocitrate. How is this going to work? So we have another enzyme called aconitase. Now, aconitase is going to form isocitrate. Before I keep going, there's a little mnemonic that people, use to remember this Tca cycle. I'll write it down for you guys. It's, so, C for citrate, so C I for isocitrate, keep for alpha ketoglutarate, that's going to be the next intermediate, S for selling, for succinyl CoA, sex for succinate, 4 for fumarate, F for 4, money, M for malate, and then officer, so to the police officer, I guess, is oxaloacetate. I don't know, I found this on Google. Guess it helps people remember it. It's very popular. But now, so we have this C for citrate going to isocitrate via aconitase. Isocitrate is now it is inhibited by a high level of ATP. Just know that. But for now, we're going to have isocitrate going to alpha ketoglutarate. How are we going to form this? We're going to use isocitrate dehydrogenase. Dehydrogenase, what? NADP, NADH, and we're going to lose the CO2, carbon dioxide. Now, alpha ketoglutarate is going to go to succinyl CoA via the enzyme alpha ketoglutarate dehydrogenase. NAD plus, NADH, dehydrogenase. Good. Succinyl CoA is now going to go to succinate via succinyl CoA dehydrogenase. Okay? GDP to GTP. Okay, so I've been saying NAD to NADH, right? These NAD and NADHs are going to be incorporated in the electron transport chain to form ATPs. Now, this is dehydrogenase, but it's not using NAD and NADH. No, it's going to use substrate level phosphorylation and go from GDP to GTP. Then, from GTP, ADP is going to join with it and form ATP for energy, and then that phosphate is going to go back up to GDP and just keep getting recycled. Not too relevant, but just wanted to mention it. Now we have succinate. Succinate goes to fumarate via succinate dehydrogenase. Dehydrogenase, we have FAD to FADH. Same thing, it's going to be involved in the ETC, electron transport chain. Fumarate is now going to go to malate via fumarase. That's the enzyme. Now this is where things get important. Malate is going to go to oxaloacetate via malate dehydrogenase. So NAD plus to NADH. Oxaloacetate gets reincorporated with Acetyl Coa via citrate synthesis to form citrate. This cycle keeps spinning, right? They call it the Tca Cycle, Krebs Cycle. It's just a while. It's like spinning. What does this mean? So we're going to form three NADHs. So 3 sets of that. We're going to form 1 FADH. So 1 FADH2. And then we're going to form 1 GTP. Okay, so now, for every 3 NADHs in the electron transport chain down here, you're going to generate 3 ATPs. Okay, so for every FADH2, since it's 2, I'm just going to make it easier. I'm going to write 2 up there. For every FADH you're going to generate 2 ATPs. And then for every GTP you have, you're going to form 1 ATP. So that means every spin of the electron transport chain is going to generate how many ATPs? Well, 3 ATPs for NADH times 3 would be 9. Plus 2 ATPs times 2 FADHs would be 4. Plus 1 ATP is going to be 14 ATPs. Just in this Tca cycle stage. Obviously, it's a lot different. How is this going to be a lot different? Well, you're going to see we have an acetyl CoA here, which is going to make NADH. In glycolysis, we have NADHs, right? They say it's anywhere from 28 to 30 ATPs for every spin in the Tca cycle. What else is important? How did I get this 28? Well, if I have 14, right? That means I really have 28 because, as you know in glycolysis, when fructose 1,6-bisphosphate splits into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, This DHAP gets converted into this. So now you have 2 molecules. So for the rest of the reaction, you have 2 pyruvates, 2 acetyl-CoAs. That means you have 2 acetyl-CoAs, 2 citrates, 2 isocitrates. So you have to multiply everything by 2. That's how we get our 28 ATPs. And where do we add the extra 2 from? Our net glycolysis reaction forms how many ATPs? 2. Okay, so we have a total of 30. I know that was on a little sidetrack, but whatever. Just know, most important thing for this video is this Tca cycle is going to form this oxaloacetate and malate. Okay, so gluconeogenesis, right? I've mentioned it 50 times and I haven't told you what it was. Here we go. Basically, all it is say our body, right, we're low on energy. And what do I mean by this? Well, if we're low on energy, we're going to be using the Krebs cycle, which is the Tca cycle. And we're going to also use the Cori cycle, which I know, this is nuts. But the Cori cycle is basically, and the Tca cycle are going to be used when we're low blood glucose. If we're low blood glucose, what are we in danger of? Yeah, we're not going to have immediate energy, so we're going to be fatigued and sluggish. But the most important thing is that our brain primarily relies on glucose for energy. It also uses ketones, but it's not healthy for us. Not as healthy for us. Glucose is the main important one. So now, how are we going to make more glucose to go into our blood? Because how does glucose get to our brain? Via the blood. Glucose passes through the blood-brain barrier in our brain, and our brain is going to absorb it and use it for energy. Now, what is a good amount of glucose that our body should have? This is just a side fact that I know and I would like to share. But in our blood, we should be at about 5 grams of glucose, right? About 5 grams of glucose in our blood at all times. Our liver should hold about 95 grams of glucose at all times. And then our muscles are going to hold 400 grams of glucose. So our body should have 500 grams of glucose stored in our body at all times. Which, if you do the math, for 500 grams at 4 calories a gram is going to equate to about 2,000 calories of carbohydrates. Which is our what? Our most effective form of energy. Why is this so important? Okay. You're fasting for a day, right? You're out in the woods, like in evolution, like back then, like when we were like cavemen. They're out in the woods, they have no food. Well, they still were able to make glucose via gluconeogenesis to have glucose go to their brains for the whole day of survival, right? They were still able to make more glucose molecules. Now, say they couldn't find food for 2 days. Then they were going to go into ketoacidosis, which they were going to make ketones, and then our ketones were going to be used for their brain. We're going to get to there in a little bit. I just wanted to mention this gluconeogenesis process is making new glucose. Gluco for glucose, neo for new, genesis, creation. Creating new glucose. For what? The brain. And how's it going to work? So it's basically, think of glycolysis in reverse with a little different substrates. So the first thing that's going to happen, right? I'm going to keep going back to my example of working out, right? I'm going to use that a lot. So you were working out, you were running for over a minute, you start to use up all your immediate glucose stores, and you go into the Tca cycle. Now, how does the Tca cycle work? We know pyruvate comes into the cycle, we make acetyl Coa. Acetyl Coa goes to citrate, all these enzymes which we're going to get to. But now, eventually, we're going to end up at oxaloacetate. Pause. Okay, so now we spoke about the whole Tca cycle, and we started getting into a little bit of gluconeogenesis. Now, to continue with the gluconeogenesis topic, how do we get our first reactant? So what's our first product? So we're going to we know glycolysis is somewhat reversible, right? But there's 3 steps that aren't. Phosphoenolpyruvate to pyruvate is irreversible. Fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate is irreversible. Glucose to glucose 1-phosphate is irreversible. So we have to have buffers, right? We need things to help. Okay, if I'm going backwards from pyruvate to glucose and there's an irreversible step, I'm like, buddy, come help me out. Get me past this step, I'll keep going. Someone else, help me out. Get me past this step, and I'll keep climbing up. Okay. What are these helpers or these steps? So, Tca cycle, right? We're going to make new glucose when what? Energy stores are low. That means glucose is low. If glucose is low, what are we using though? We're still using this Tca cycle. How? Fat and amino acids. So if this Tca cycle is still pumping, right? He's still chugging along. He's going to make our first substrate or intermediate that's going to be involved in glycolysis. How? So, oxaloacetate, right, is then going to be reconverted into malate. Okay. Malate is going to become oxaloacetate now in the liver. Where does the Tca cycle occur? The mitochondria. That's why the mitochondria is the powerhouse of the cell, right? Makes 30 ATPs, powerhouse. Malate goes to oxaloacetate in the liver now. Oxaloacetate is going to go to phosphoenolpyruvate. Where? In the liver. Via Tca. Tca is going to be our main enzyme. Tca is known as phosphoenolpyruvate carboxylase. Carboxykinase, I apologize. Now, see how I took it from the Tca cycle? Let me run through it from the top. Pyruvate becomes oxaloacetate. How? Pyruvate carboxylase, right? Oxaloacetate can become phosphoenolpyruvate via Tca, phosphoenolpyruvate carboxykinase. Now, didn't I just say pyruvate goes through the whole Tca cycle and then forms this oxaloacetate? Yes. But now, if we have pyruvate, right, laying around that we can't use it, it's going to travel, or an enzyme known as pyruvate carboxylase is going to turn it into oxaloacetate. So oxaloacetate goes to the malate, right? That malate goes back to oxaloacetate, etc. I'm not saying again. And it becomes phosphoenolpyruvate. Phosphoenolpyruvate gets incorporated where? Right here in this is our glycolysis pathway, but now it's our gluconeogenesis pathway. So boom, we had stuff going to pyruvate, and now like, oh, we're stuck. We can't go backwards. Pyruvate's gonna come here, turn into oxaloacetate, go to PEP. PEP's like, all right, I can go up now. Phosphoenolpyruvate's flying up, right? He goes to 2PG, 3PG, 1,3-BPG, glyceraldehyde 3-phosphate. He's like, oh, I can't go to fructose 1,6-phosphate, bisphosphate. What am I going to do now? No need to fear. Here we go. They're going to swarm up into fructose 6-phosphate via another reaction. What's this reaction? Okay. Fructose 1,6-bisphosphatase, which is different than fructose 1,6-bisphosphate, so now it's the enzyme for it, is going to take glyceraldehyde 3-phosphate or dihydroxyacetone phosphate, and it's going to go up to fructose 6-phosphate. So it's going to skip this step. Well, who tells fructose 1,6-bisphosphatase to start? Like, who's like, whoa, why am I even doing this? Like, who told you to turn me on? Okay. Cortisol and glucagon are going to stimulate 3 different enzymes. Mainly cortisol. Cortisol is going to stimulate fructose 1,6, pyruvate carboxylase, and Tca. Glucagon is mainly going to stimulate pyruvate carboxylase and Tca. But just know, cortisol and glucagon are going to stimulate gluconeogenesis. Why? If you're stressed, right? If you haven't eaten in a day, you're going to be a little anxious, right? You're irritated, you're stressed out. Okay? Glucagon is what? The opposite of insulin. Insulin takes sugar into our body. Glucagon does what? Helps bring substances to make new glucose. Now, if we have high cortisol and high glucagon, that means we have a high Tca cycle. If we have a high Tca cycle, we're going to eventually come to a point where we're making too much citrate. If we're making too much citrate, this citrate can turn into fat. However, if we have a lot of acetyl-CoA with this step, the acetyl-CoA is going to do what? This acetyl-CoA is going to also stimulate pyruvate carboxylase to make PEP. Now, inhibitors are going to be ADP and CoA. What does that mean? If we have low energy like ADP, we don't need to stimulate pyruvate carboxylase. So how are we going to increase that energy? Tca cycle, baby. He's got to start it up. So now, Glyceraldehyde 3-phosphate, dihydroxy acetone phosphate. Skip over, go to fructose 6-phosphate. Fructose 6-phosphate is like, I'm going up to glucose 6-phosphate. And then he's like, all right, I'm going to travel up. I'm going to keep traveling up. No. Irreversible steps come in. So, how do we go to glucose 1-phosphate, or to glucose, right? Glucose 6-phosphate enters into this orange region. This is our smooth ER, smooth endoplasmic reticulum. This G6P then travels down and it gets hit by something. It's known as glucose 6-phosphatase. He's going to, I'm taking off the phosphate. So all it does is takes off one of the phosphates, and now we have glucose. So glucose in the smooth endoplasmic reticulum is then going to come out of the smooth ER. It's going to travel straight up into the blood. He travels into the blood, go to the brain, we're moving, right? Gluconeogenesis is started. Now, what else can happen? So I said blood glucose levels are low. So we're in a hypoglycemic state if we're using gluconeogenesis. The primary role or energy is the brain. A secondary form of energy for glucose, or not for glucose, but for our brain is ketone bodies. What are ketone bodies? And how are they involved in gluconeogenesis? They are a little bit. So, we're going to have a process called lipolysis, which is going to split a fatty acid molecule. So this glycerol and these fatty acids are going to split. And the fatty acids are going to travel out. We're going to talk about them later. But the fatty acids are going to travel, and then this glycerol is going to be converted into dihydroxy acetone phosphate. Whoa. What? So DHAP becomes, same here, right? Glycerol kinase takes it and forms DHAP. DHAP comes back around the rate of fructose 1,6-bisphosphatase, stimulated by cortisol and glucagon, travels fructose 6-phosphate. Fructose 6-phosphate travels up to glucose 6-phosphate, back into the smooth ER, glucose 6-phosphatase to glucose, out, up, into the blood. So boom, now the two different ways. So far, for gluconeogenesis, we had one, excess buildup of Tca cycle forming oxaloacetate, and two, breaking down some fatty acids. Those are the two main ways. Got a couple other ways, of course. So now talk about that. Muscle. I know I said liver, but now we're going to talk about muscle real quick, because what did I say? Fat, pyruvate, right? Can go to gluconeogenesis. How can my muscle form glucose? Because didn't I say 400 grams are stored in the muscle? I did. Okay. Muscle, right? So we have glycogen in our muscle, right? We know this. Glycogen, let me write, about 400 grams if you're fully, you're a bum all day, you're not doing anything, you don't go to the gym, you don't burn it. I shouldn't say bum, but you're not burning. So glycogen can get converted into glucose 1-phosphate via muscle glycogen phosphorylase. Okay, so similar to glycogen phosphorylase, but it's in the muscle, so muscle glycogen phosphorylase. Now this glucose 1-phosphate can turn into glucose 6-phosphate via phosphoglucomutase. Same exact thing, glucose 1-phosphate, glucose 6-phosphate via phosphoglucomutase. See how everything's like overlapping? This glucose 6-phosphate, now, in the muscle, what do we use? Hexokinase. If we're in the liver, what do we use? Glucokinase. I wrote hexo just to tell you guys before, but I'm going to erase it now. Liver gluco, muscle hexo. Cool. So now, glucose 6-phosphate, two different cycles. We're going to have the glucose-alanine cycle, and we're going to have the Cori cycle. We're going to talk about the Cori cycle first. So the Cori cycle, right? We know glucose 6-phosphate goes to glycolysis. You make 2 ATPs, form pyruvate. Pyruvate can then turn into lactic acid. So I'm running, right? Or I'm lifting heavy weights. I'm burning, right? Eventually I'm going to run out of, what? The 45 seconds of working out, I made pyruvate. Pyruvate is then going to turn into lactic acid in the muscles via lactate dehydrogenase. Oh. Dehydrogenase again, NADH to NAD+. Now, this lactic acid in the muscle, right? Why do we even use lactic acid? What's the point of it? The end goal is to get pyruvate from the muscle into the liver. However, pyruvate cannot travel through the blood-brain barrier. Lactic acid can, so it's created. So, pyruvate becomes lactic acid, the lactic acid in the muscle is going to travel out of the muscle into the liver. So we're going to have lactic acid in the liver here. This lactic acid is going to then travel up into pyruvate. How? Same thing. Lactate dehydrogenase now, but this time we're going to go from NAD plus to NADH. So we're going backwards, right? Pyruvate is then going to go up to G6P. What is G6P? Glucose 6-phosphate. Glucose 6-phosphate. How did it even get to G6P? You guys got to know all the steps. The pyruvate then becomes phospholinopyruvate. All the way up here, G3P DHAP, fructose 6P into glucose 6P. Same thing. Glucose 6-phosphate travels into the smooth endoplasmic reticulum. Glucose 6-phosphatase cleaves off of phosphate. Glucose travels out, up blood-brain barrier. New glucose. Now glucose is elevated. Glucose can travel back down via hexokinase for the muscle. Glucose 6-phosphate. Process can keep going, right? That's why they said lactic acid is kind of good for you. You're running, right? You're building lactic acid. Well, that lactic acid is going to be used for energy. Now, the glucose-alanine cycle is a little different. Same end result, though. Pyruvate, right? So same thing, glycolysis, 2 ATPs, etc., is now going to mix with an amino acid known as glutamate. When it mixes with glutamate, it's going to form alanine in the muscle, and then alpha-ketoglutarate. Where is alpha-ketoglutarate? Right here in Tca cycle. So this alpha-ketoglutarate is going to go into the Tca cycle intermediate for the Krebs cycle, right? Oxidative phosphorylation. Now, the alanine in the muscle is going to come out, and the alanine is going to be in the liver now. Why did we do this? Why did we make alanine in the muscle, alanine in the liver? The end goal is what? Pyruvate, but pyruvate can't travel through the blood-brain barrier. I don't know why, that's just what it is. So pyruvate and glutamate form this alanine. Alanine passes through. Alpha-ketoglutarate comes back. Right there, he comes back. This alpha-ketoglutarate makes two different things. Pyruvate and glutamate in the liver now. Now we have the same work. The ends justify the means. Everything's full circle. We started as pyruvate and glutamate. We're in pyruvate and glutamate again. Pyruvate travels up. Well, this whole process of gluconeogenesis, you know where it's going. Traveling G6P, SCR, glucose, blood, brain, right? Well, what happens to this glutamate, right? We can't just have it floating around in our liver. It's dangerous. He's going to be broken down into alpha-ketoglutarate again and into NH3, which is ammonia. This NH3 is going to be involved in the Urea Cycle, which is how we're going to get excreted out of our body via the kidneys. Now, why is this going to be not the best thing for us? Because we know ammonia is toxic, especially ammonium, NH4. We're going to get to it though, but this Urea Cycle can be very toxic to our body. Where do we get a lot of nitrogen or urea? Well, in high fat, high protein, low carb diets, known as the Atkins diet. Ketoacidosis is going to be created, which we're going to get to, and it's going to literally rely on these systems, and we're going to elevate our nitrogen. But for now, just know for the rest of gluconeogenesis, those are the main pathways to get new glucose into our brain for our primary supply of energy. Okay, so we just covered how the monosaccharides go through. Right now we talked about gluconeogenesis, forming new glucose. Well, we're in a state, right? Too much ATP. I went to McDonald's, right? Had 3 burgers, 50 chicken nuggets, 4 large fries, just grubbed out. I have way too much carbs, way too much protein, way too much fat. I'm going to use some of it, but now if our ATP, right, too much, we're going to form fat. How are we going to form fat? All right, so we're going to take it from glucose. Originally, right, I had, let's say my liver, right? I could store 95 grams of carbs, we said, which equates to 380 calories, right? 95 times 4, 380. 380 calories, so 95 grams. I had 2 large Diet Cokes at McDonald's, which is about 70 grams of carbs. And then say I had 4 ice creams, right? Because I was just feeling the ice cream. I'm way over my sugar balance. So I go through glucose, right? I refuel my whole entire liver. Liver's fueled, right? It's filled up. Well, some of it's going to go through Tca Cycle, the Krebs Cycle, and then we got to use it for energy. But now, right, we're pumping Acetyl Coa, right? Pumping citrate, pumping isocitrate. What happens? I said it before in the beginning. ATP, or high ATP, inhibits Isocitrate dehydrogenase. So it inhibits the conversion of isocitrate to alpha-ketoglutarate. Okay, so if this is not occurring, right? If we can't make alpha-ketoglutarate, well, that means isocitrate is stuck, right? It's a reversible reaction. So Aconitase is like, all right, screw this. I'm going to make citrate again. So now we have a lot of citrate, right? Citrate is just building up. Boom, 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 boom. Citrate's building up. What happens when citrate builds up? Okay? So we're going to start making fat. It's going to travel up, and then this thing called citrate lyase. So citrate synthase went from oxaloacetate and acetyl CoA into citrate. Citrate synthase. What do you think citrate lyase does? Well, synthase is to make, lyase is to cut, right? So it's going to cut, it's going to add a CoA, and it's going to cut it. It's going to cut the citrate. So it's going to form oxaloacetate and then acetyl CoA. This CoA is being added to an acetyl group here, just like it's being removed here. And we have oxaloacetate and acetyl CoA. What can the oxaloacetate do? Well, oxaloacetate can go to malate. Malate, what? Oxaloacetate can go to malate. Malate can then go to pyruvate via a malic enzyme via NADP plus to NADPH, which I said was super important in fatty acid synthesis, which is going to be used. I'll put an asterisk here. And this pyruvate can do what? Well, instead of going directly back into acetyl CoA for the Tca cycle, it can go from the oxaloacetate malate, gluconeogenesis shuttle. Now, if this is occurring, right, we're saying we're using lots and lots of oxaloacetate going to malate. Well, that means oxaloacetate eventually is going to be reduced. So if oxaloacetate is low, we can't form any more acetyl CoA, so we can't form any more citrate, right? Not acetyl CoA. We can't mix oxaloacetate and acetyl CoA to form citrate. So by reducing oxaloacetate, we're going to end up reducing citrate formation. Okay, so if we reduce the citrate formation, we'll end up reducing the fatty acid synthesis, thankfully, because we're going to be freaking marshmallows, like just walking around. However, this acetyl CoA, it's then going to be used in a lot of different reactions, which is going to be our ketone metabolism, which is for ketosis, which we'll speak about after. But just note, the decrease in oxaloacetate is going to end up decreasing citrate. But I said this thing split into 2. So one was oxaloacetate, one is acetyl CoA. This acetyl CoA in this state, right? Not in this state, in this state. It's going to be activated, or not activated. We're going to have another enzyme that's going to form malonyl CoA. This enzyme is going to be known as Acetyl Coa Carboxylase, right? ACC. So it's on Miami or Virginia Tech or Louisville, right? All our ACC schools. Acetyl Coa Carboxylase is going to form malonyl CoA. There's going to be 2 things that stimulate it, and there's going to be some things that inhibit it, just because that's what we speak about in biochem. Allosteric stimulator is going to be citrate. Well, that makes sense. If we have citrate, we're going to make malonyl CoA. And the next one's going to be insulin. Well, that also makes sense. I just had a huge thing of McDanks, right? All that soda, all those fries. McDonald's is what I mean when I say McDanks. All those fries, right? So insulin's released, making a lot of malonyl CoA. What's going to inhibit it? Well, long-chain fatty acid CoAs, cortisol, so if we're starving, glucagon, epinephrine, norepinephrine, right? What are those guys? Catecholamines used in our fight or flight, running away from a bear. If we're running from a bear, who gives a shit? Who cares if we're making lots of fats, right? What do we want to do if we're running away from a bear? We just want to have energy and want to start going. So it's going to inhibit this. Now, malonyl CoA has 2 different fates. What is it? Well, it doesn't even have two different fates. It's going to inhibit something. It's going to inhibit the process of CPT1. Okay, where am I? Where is this? Remember the mitochondria, right? We have carnitine, palmityl, transferase 1 and 2. These things take long-chain fatty acids into the mitochondria to break down the fat to make what? Energy. Well, we want to inhibit this because we don't want to break down fat. We want to make fat. Okay, so if we're inhibiting this process, what are we going to start to activate? Well, we're going to have malonyl-CoA involved in our fatty acid synthesis. Now, how does this ACC, what regulates him? Well, we said allosteric and hormonal, but something else is going to regulate it. These cells are usually in their inactive stage, right? If citrate comes, he's going to activate what? He's going to activate our Acetyl Coa Carboxylase. Insulin's going to activate this. All these things are going to end up activating it. How? By putting these cells together, right? They're grouping them together. When all this stuff is grouped together, we're going to form something. We're going to form FAS1, fatty acid synthesis type 1. So now, in our formation of fatty acid synthesis, we're going to have FAS1, we're going to have malonyl-CoA, and we're going to have NADPH. These three things are going to have our formation of fatty acids. Fatty acids are then going to be used for something else. Now, we have our fatty acids, right? Where are we going to go? Where are they going to go now? Triglyceride synthesis. Well, what's the difference? What's a triglyceride? Triglyceride is a glycerol and fatty acid tails, our main form of fat. So our fatty acids, 1, 2, 3 of them, and then we're going to have our glycerol. Where's our glycerol going to come from? Had two huge sodas, all that ice cream, all this extra glucose goes through a GLU2. Glycolysis, we're going to form DHAP. Where is DHAP? I've spoke about him a lot. DHAP is in our glycolysis pathway, dihydroxy acetone phosphate. Okay, so we have either glyceraldehyde 3-phosphate or DHAP. DHAP is going to be used, and it's going to form glycerol 3-phosphate via glycerol kinase. Glycerol kinase is going to take NADH and make an NAD+, right? Not too important right now, but just know glycerol 3-phosphate is then going to form our glycerol molecule. How else can we form our glycerol molecule? Well, adipose tissue can travel through lipolysis and then be converted into glycerol via glycerol 3-phosphate dehydrogenase. Okay, so that would also be an NADH to an NAD. Okay, actually NAD to NADH, I apologize. Okay. Where did the adipose tissue get the fat from? VLDLs. Where did the VLDLs get the fat from? Lipoproteins. Where did the lipoproteins come from? That's what we're going to talk about now. So it's this whole chain reaction. Now, these fatty acids are going to form with glycerol via acyl transferase. It's going to happen 3 times. So 3 different times this enzyme is going to work. Why 3 times? Well, the first time it's going to look like this. It's going to be our glycerol with 1 fatty acid tail. Second time, glycerol with 2 fatty acid tails. Third time, 3 fatty acid tails. So this is a MAG, monoacyl glycerol. This is a DAG, diacyl glycerol. And this is our TAG, triacyl glycerol, right? Okay. Now, triacyl glycerol can go 2 ways, right? It can go into a lipoprotein, which can become a VLDL, which then can go into our adipose. Or, more importantly, we're going to have to mobilize these fats, right? Because we don't want just fats floating around in our body. We're going to have to put it in our bloodstream. We're going to have to put it in other ways. So here we go. Mobilization of fats. When are we mobilizing fats? So this is how we made them. How are we going to mobilize them? How are we going to use them? A fasting state, right? So our blood glucose levels are low. If our blood glucose levels are low, we have a thing called the pancreas, right? He's going to release 2 different types of cells. He has, these pancreatic cells are known as A cells and I cells. I cells are for insulin, A cells are for glucagon. He's going to release glucagon. Then our adrenal medulla is going to have chromaffin cells, which is going to release epinephrine and norepinephrine. Both of these catecholamines are going to be used as well. Glucagon, epinephrine, and norepinephrine, if you remember from our G-coupled protein receptor videos stimulate GS. GS is a messenger, right, that stimulates PKA, protein kinase A. Protein kinase A is then going to phosphorylate two different things. We have this thing in baby blue called paralipin, and then in orange is going to be known as CGI. So PKA is going to phosphorylate it. Now, when the PKA phosphorylates it, CGIs are going to target what? A triacylglyceride, so a tag. Step 1. Adipose triglyceride lipase, or ATGL, is going to come and cleave off one of these fatty acid tails. It's only working when CGI is phosphorylated. That's step 1. Step 2. Hormone-sensitive lipase, HSL, is going to cleave off the second tail. So now we had a diacylglyceride, or we went from triacylglyceride to a diacylglyceride. Cleaved off another fatty acid tail. Okay? Step 3, monoglycerol lipase, or monoacylglycerol lipase, is going to cleave off the third fatty acid tail. When it cleaves off this third fatty acid tail, we're going to be resulted with a fatty acid and a glycerol. So now, we have 3 fatty acid tails, free fatty acids, I should say, and a glycerol. The glycerol can be diffused right into the blood, travel up, be converted into glucose, and goes where? That glycerol comes into gluconeogenesis. Cool. What happens to the 3 fatty acids? Well, a protein called albumin in the liver takes it up, puts it into the blood, sends it through beta oxidation. I'm not going to talk about beta oxidation. I've talked about it a lot in the past. But our beta oxidation now is going to be our main thing involved in our ketone metabolism, right? Everyone's favorite thing, the Atkins diet, right? Everyone loves this. Okay. So now I'm going to film this part, and I'm going to talk more detailed into the Atkins diet, mainly because my mom's filming it and she wants to hop on the diet, which I don't think is the best bet, but here we go. Ketone metabolism, right? When does this occur? When your blood glucose is low. Pause it. Whatever. When does ketone metabolism take place? Well, it's in the Atkins or the keto diet, of course, like I said. It's also when our blood glucose levels are low. So we don't have a lot of carbs, we don't have a lot of sugars. Prolonged starvation, haven't eaten in a while, right, cavemen? And uncontrolled diabetes, specifically type 1. Why? Well, if they can't control their diabetes, what is the difference between type 1 and type 2? Type 1, they can't create insulin. So they have to give themselves insulin. They have to inject it. Type 2, they could release insulin, just not a lot of it. So if you can't control your diabetes, specifically type 1, and you don't give yourself insulin, say, you're going to have no blood glucose in your body. Your blood sugar, when they test for blood sugar, they're testing the blood to see if it's at that 5 grams, your A1C. Now, if it's down, right? If it's like, oh boy, we're in trouble here. Our body's not just going to like say we're dead, right? It's not going to, we have a secondary mechanism, ketones. Primary glucose, secondary ketones. Okay, so these ketones, we're going to form ketones to travel to our brain. And how are we going to form them? This is how we do it. First, we said we created a lot of fat, right? Well, what happens, what occurred when I said we did gluconeogenesis, right? We kept doing gluconeogenesis over and over and over again. So oxaloacetate came to malate. Malate shot across, glucose into the body. We rev that system up, right? We rev that sucker up. Well, oxaloacetate is going to drop. We're going to have low oxaloacetate. Low oxaloacetate means 2 things. Can't make a lot of citrate, or can't make more malate. Can't make more malate, can't make more glucose. Okay, what are you making? If you can't make more citrate, well, because oxaloacetate and acetyl CoA combine to form citrate, acetyl CoA is just going to be left. It's just going to be in the body, free floating, right? Is this healthy? No. So we can't make citrate for fats. We can't make acetyl CoA be used anywhere else. We're going to send it into this system right here. So the oxaloacetate is decreased from malate, going to gluconeogenesis, right? Can't join with acetyl CoA, no citrate. Acetyl CoA, we're going to have a lot of them. A lot of them. They're coming from this region and from beta oxidation. So we have a lot of them. 2 of them are going to join together, and we're going to form acetyl acetyl CoA. So CoA is going to come off. So two acetyl CoAs join together, loses a CoA via acetyl CoA acyl transferase, and we're going to have aceto for an aceto without a CoA on it, and acetyl CoA. Now, we're then going to join another acetyl CoA because we had so many of them. This acetyl CoA and acetyl CoA join together, loses another CoA, and forms HMG CoA, which is HMG CoA synthase, is the enzyme that does it. This is our rate limiting step, right? Very important. Now, unfortunately, I don't know why the body does this, but we're going to now lose an acetyl CoA. We've been gaining them, now we're going to lose them. HMG CoA lyase is going to lose an acetyl CoA, and we're going to form acetoacetate. This is where things get important. Acetoacetate can go one of two ways. Three ways, right? But acetoacetate is our first ketone. So it's going to be one of our first ketones we're going to use in our body. For what? The brain. For energy. So, you're on one of these diets, right? You're not eating any carbs. If you're not eating any carbs, you're never making this glycolysis pathway work. Why? Because you can't put glucose into the body. Are you even using the Tca cycle? A little bit, right? Via amino acids and stuff. But you're not even using this cycle. You're getting your energy from the breakdown over here. There's your your energy is low, right? So now, one of the ketones that's going to supply our brain is going to be acetoacetate. Next, beta hydroxy butyrate dehydrogenase is going to be, it's going to take an NAD to an NADH, right? And we're going to form beta hydroxy butyrate. Well, we know a dehydrogenase makes 3 ATPs, right, with the NADH. So it's going to be a little bit of energy. And this is going to be our secondary ketone. Now say, right, we're in a state where we're just making so much of these guys, more and more and more. Eventually, we're going to throw up. We're going to start vomiting. Why? Ketones are acidic. Their pKa is around 3.5, I'm pretty sure. This is acidic, you're going to start vomiting. Now, say you don't vomit, right? And you're just going through ketoacidosis, ketones, right? You're on this diet. Acetoacetate decarboxylate is going to form acetone. Okay, this acetone is why you have alcohol breath. So if a patient comes in, right, mainly, this is how you could diagnose if someone's a type 1 diabetic. Patient comes in, they're very lethargic, they're very tired, right? And their breath smells like they just came from the bar and were hammering shots all day. It's because they have acetone breath, which is alcohol. Why would it smell like alcohol? Well, the type 1 diabetic cannot ingest or release insulin, so no glucose, even if they ate glucose, even if they ate McDanks. They can't get any glucose into their blood because insulin wasn't secreted. That insulin, right, since it's off, we couldn't make glucose to go to our brain, so we made ketones. Excess ketones get converted into acetone via acetoacetate decarboxylase. So they're going to be tired, lethargic, right? They're going to smell like alcohol. They could even be vomiting excessively because we're making so many different ketones. All these ketones are very acidic to our body. Yes, they keep us alive in our brain, but they come with consequences. Now, another thing. How do we break these things? How do we break this down, right? Because we just don't want free-floating ketones. Like, it's not too good. We're going to go into our muscles, right? Our muscles are like, all right, I'll help you guys out. I mean, I don't like it, but I'll help you out. So we're going to start from the bottom now because this is step 1, this is step 2. Acetoacetone was 1. Beta hydroxybutyrate is 2. This whole process is reversible, thankfully. So we're going to start off in beta hydroxybutyrate. He's going to get converted into acetoacetate. So same thing. We came from here, came up. Beta hydroxybutyrate down to acetoacetate now. Via beta hydroxybutyrate dehydrogenase. Okay, same enzyme. NAD plus to NADH. ATPs, you know this, ETC, electron transfer chain. Acetoacetate is then going to form acetyl CoA via thiophosphorase. What is cool about this guy? Well, this guy gives off or receives a CoA to form acetyl CoA. How? Tca cycle, succinyl CoA, right, becomes succinate. When this happens, what occurs? How does succinyl CoA go to succinate? Well, the GDP and GTP occur, Succinyl CoA dehydrogenase, we lose a CoA. When we lose that CoA, we use it in this reaction, and we form aceto-acetyl CoA. Cool. This aceto-acetyl CoA could go two ways now. Aceto-acetyl CoA could go into acetyl CoA via acetyl CoA Acyl Transferase. So we're giving it another CoA. Now, we form two acetyl CoAs. That's the end result. Because why? Aceto, so one's an acetyl CoA, one's not. But we added that CoA to it. So now we have two acetyl CoAs going to the electron transport chain, forming ATP. What is very dangerous about this step? Well, this is how we break it down, right? Now, we have something known as the Urea Cycle. Now, what is this? We have so much acetyl CoA, right? Acetyl CoA came from where? The breakdown of these ketones. If we're making a lot of it, what else are we making a lot of? Alpha-ketoglutarate, Tca Cycle. Okay. Now, transamination and oxidative deamination we didn't really talk about too much, but if we have a lot of alpha-ketoglutarate, a lot of alanine, it's just an amino acid, where is these amino acids coming from? If we're on that ketone diet, amino acids come from proteins. Proteins, what? What are the 20 main amino acids from proteins? Isoleucine, leucine, valine are the branch chain amino acids, right? The essential, and then we have the non-essential ones: valine, phenylalanine, isoleucine, tryptophan, leucine, methionine, threonine, histidine. Those are essential. And then the ones that we make in our body are alanine, right? That's a non-essential. So if we have all of these amino acids from the excess protein, and then all this alpha-ketoglutarate from the Tca cycle, we still got to break them down. ALT, which is alanine aminotransferase, is going to make pyruvate and glutamate. Okay. Glutamate is then going to be turned into, should I write that? So glutamate again, right, is going to leave. It's going to be glutamate, and then glutamate dehydrogenase is going to turn it back into alpha-ketoglutarate. So why is this happening? Well, we're going from the muscles, right? We're in the muscles, back into the liver. So now we're back into the liver. And then we're going into the mitochondria. So this is now we're in the mitochondria, the alpha-ketoglutarate. Alpha-ketoglutarate, well, this glutamate makes alpha-ketoglutarate and NH3, which is nitrogen. Ammonia, I should have said. Via glutamate dehydrogenase. It goes from NAD plus to NADH, and H2O is added. This NH3 plus, right, for the ammonia pathway, is then going to form with a free radical hydrogen. So another free radical. We're going to form ammonium. This ammonium, right, is from excessive protein breakdown. Where else do we see this occurring? Bodybuilders, right? They eat 300 grams of protein a day. It's pretty dangerous. Why? This ammonium is going to leave the body and it's going to go to the blood-brain barrier. Now, what's the most important thing we know about the brain? In the blood-brain barrier, we have something called astrocytes. These astrocytes, right, are a type of cell that's going to start taking glutamine. Well, it's going to take glutamate, and it's going to form glutamine via glutamate synthase. Now, we have glutamine, right? What else does it form? It also forms glycine. Okay, you're like, cool, I have glutamate and glycine in my, or glutamine and glycine in my brain. Why does that matter? What do these things do? These guys suck water into the brain. They absorb water into our brain. What does that mean? We're going to cause cerebral edema, also known as brain swelling, which increases our intracranial pressure. If we increase our brain swelling, cause an increase in intracranial pressure, we're going to have aneurysms in our brain. We're going to form brain aneurysms, right? We have so much water and blood in our brain, so much intracranial pressure, they're going to have to do a bury hole, right? They're going to have to put a drill into our brain to make a hole to relieve all this pressure. Where did this come from? This came from the excessive ammonia that was turned into glutamine or glycine via astrocytes in our brain. So we're going to have brain aneurysms, and unfortunately, the outcomes are usually not too good. So now, what else can happen? So that's the ammonium. Now here we go. We also made alpha-ketoglutarate. The ammonium, if it doesn't go to the brain, where does it go? This ammonium in the mitochondria how do we break it down? The safe way, when there's not an excessive amount. We add 2 bicarbonates, HCO3s, and 1 ATP, and we're going to form carbonyl phosphate, which is CPS1, carbonyl phosphate synthase type 1. Carbonyl phosphate is then going to combine with ornithine. When they combine, they're going to use something known as ornithine transcarbamylase, right? That's going to form citrulline out of the mitochondria. Okay, so it's going to form citrulline. Now, in the malate aspartate shuttle, we have malate and aspartate. This aspartate is going to mix with citrulline, and it's going to form arginosuccinate via arginosuccinate synthetase. This arginosuccinate is then going to go via an enzyme known as arginosuccinase enzyme is going to make fumarate and arginine. Okay? Fumarate is then going to be used where? Tca cycle. Where is this arginine going to be used? Arginine is another amino acid, we know. Arginine is then going to be converted into 2 things, ornithine or urea via arginase. This urea is then going to be safely excreted out into the kidney, and then this NH3, ammonia, is going to be filtered out through our body. We know NH3 is not as dangerous as NH4. NH4 ammonium reacts with glutamine synthetase to form glutamine and glycine in our brain, which is dangerous. The NH3 is not as dangerous as NH4. Okay, so in this video, we spoke about all the different ways the liver reacts for our macromolecules, our fats, our proteins, our carbohydrates. So I know that was a long one, and I hope it helped.